these cells of the nervous system called neurons, as we start to get into how they work, it's going to start looking awfully complicated, but oddly familiar to what we've talked about with muscle. That's why I wanted to rush on into this stuff this week. So let's talk about how the whole nervous system is organized. This is your body's major long-distance signaling system, communicating from one part of the body to the other. And I am going to try to imitate. If you guys haven't watched Dr. Schultz's wonderful video on this part yet, I'm going to try to imitate her super cool drawing that she does, where she does this thing that kind of makes the nervous system look, oh wow, this is not going to, oh, that's not going to go well. Let's try that again. Where she makes it look, the whole thing look like a big ice cream cone. It's sort of funny, but awesome at the same time. It's such a great, simple way to try to sketch the central nervous system. Y'all are like, what? Okay, well, brain and the spinal cord. Brain and spinal cord. And with, excuse me, sorry. I went and got some nice toasty warm afternoon coffee, but I need to be drinking it too for my throat. My apologies. Within the brain and spinal cord are these bundles of what are called axons. And we haven't talked about these yet. We'll get to them. I got the word right over here. Axons are these microscopic parts of neurons, where we did touch on them back in the last chapter, well, back in chapter 10, I should say, as we talked about that axon of that motor fiber coming down and forming a synapse with muscle fiber. And we looked at that place right under there called the motor end plate, where it attaches to a muscle fiber. So axons don't float in from outer space like it looked like here. They're actually in these bundles like cables, like wires and cables that are bound up wires and wires and wires. And within the brain and spinal cord, these bundles of axons are referred to as tracts. Outside the brain and spinal cord, way down here in the bottom left, outside the brain and spinal cord, they're called nerves. So within the brain and spinal cord, which is referred to as the central nervous system, they're called tracts, these bundles of axons. They're sending impulses, high-speed impulses, up or down. Out in the peripheral nervous system, those bundles of axons are referred to as nerves, and the basic organs of the peripheral nervous system are those nerves, as well as these other structures we'll talk about in a few chapters from now, called ganglia, when we get over here to the, when we get to the autonomic nervous system in chapter 14, I believe it is. Let's see if we circle back around to it in chapter 14. Yeah, it's chapter 16 in our textbook. But it's right at the very end, and we'll get back to it. So actually, from this point onward, it seems like we're only about halfway through the semester. But from this point onward, it's all nervous system. We're just going to talk about different parts of the nervous system, like how your eyes work and how your ears work. But they're parts of the nervous system. They're senses. We're going to talk about the sensory systems of touch and motor response, and the brain and the spinal cord in more detail. So it's amazing. These terms that we're talking about today are going to be terms that we're going to carry through with us constantly for the rest of the semester. And even they'll come up again next semester. So we have within the brain and spinal cord these bundles of axons called tracts. Some of these tracts are tracts that go up the spinal cord, carrying impulses up the spinal cord to the brain. These are known as ascending tracts. And that's how the brain gets input from the outside world. That's how it takes in information from the outside world. But the brain sometimes needs to respond, like it needs to send impulses to skeletal muscle. So it uses different sets of axons that only send impulses down from the brain. Those are called descending tracts. Okay? Now, the brain and spinal cord also have to get input from the outside world, like from your, let's kind of make this sort of try to look like your body. I don't know why I'm making you look like a teddy bear, but from your skin, like from the surface of your skin, you need to get impulses coming in from your skin. And these are usually coming in more from these neurons, from these nerves, kind of more at a direction that's perpendicular. And these pathways are referred to as sensory pathways, also known as afferent pathways. Afferent. I try to say afferent, by the way. I try to say the A really hard because you can see the other version of the word is efferent. And I find those words are easily confused. My teachers used to say afferent and efferent. Afferent, efferent. And I'd go, excuse me? I can't tell the difference. It sounds like you're saying almost the same thing. So I try to say a really strong afferent and efferent when I'm talking about these, but I really prefer the alternate terms sensory and motor. So afferent sensory fibers coming into the spinal cord, and they will then connect with the ascending fibers. And this is sort of like a two-part pathway of receptors in your skin feel cold, so they send impulses to the spinal cord that then connect to ascending tracts up to the brain, and that's how the brain gets the notification that your skin feels cold. So the brain can then respond to that cold using these descending tracts. But remember, when you're cold, you typically will shiver. So the spinal cord has to then send impulses out from the spinal cord out to your skeletal muscle to cause the muscle to, let's just, do something like this that makes it look like shivering. Now, these fibers that are coming out of the spinal cord at a kind of a perpendicular direction to the spinal cord are those motor fibers. Those fibers that are, how did that get turned on? That needs to stay off. One of my kids been messing with my phone. I always keep my ringer sound off. But the efferent is the motor fiber. The efferent is the motor fiber. The efferent that does the effort to move. Sort of. The word efferent by itself. Oops, sorry. The microphone came off. I'm sorry if that shocked your headphones. Efferent means going away from or going out of. Afferent means going towards or running towards. But yeah, that's a way of thinking about it. Efferent, the effort to move. Mm-hmm. All right, so ascending tracts and afferent fibers are both going more into the spinal cord or up towards the brain, how the brain gets its input. The descending tract and the motor fibers are how the brain sends messages to other body parts and tells them to do their thing. Okay? So it says at the very bottom of page 131, make a diagram of sensory and motor neurons traveling through the CNS and PNS, and that's what I tried to do, modeling mine after what Dr. Schultz has in hers. So I like the way she does it a little bit easier. Okay. Top of page 132, let's take a look at how we can apply some of these terms. In the peripheral nervous system, there are sensory and motor neurons. So in PNS, the other word for sensory is afferent, and motor is efferent. All right, so in that line at the very top of 132, you should have afferent and efferent. Yeah, that's another way of thinking about it. What's interesting about the way you phrased it with the word running is that's actually what the word pharaoh kind of means in Latin. It sort of is running or going or moving in a direction of. So afferent means running towards, running at, and efferent means running away from. So yeah, you're pretty close there with your way of thinking about it. So the next line, so it says it helps to think of these neurons as horizontal, going to and from the spinal cord. So that's kind of how I tried to draw them, like this. Yeah, absolutely. Absolutely make it easy on yourself. Make up as many things as you can. In the central nervous system, there are sensory and motor neurons, right? So it looks like saying the same thing, but in the CNS, the sensories are ascending and the motor are descending. Okay, so in that third line, in the parentheses, sensory parentheses, ascending, motor descending. So notice how we can use sensory or motor in either CNS or PNS. And it kind of emphasizes that right there under functions of neurons. Sensory, afferent and ascending, motor, descending, and efferent, depending on which division of the nervous system you're talking about. But here's the thing. Let me see if I can draw a slightly different color that stands out. Okay. Within the brain, this input, that doesn't stand out very well at all. I'll just try to make it a little bit darker. This input has to be processed and integrated for the brain to determine what is the appropriate response. So a lot of what the brain and spinal cord are made up of are referred to as interneurons that do integration. Sorry if that just does not show up very well, but that's letter C there near the top of page 132. Okay, and then under terminology, nerve and tract, we kind of got that. A tract is bundles of axons in the CNS. Nerves are bundles and axons outside the CNS. But check this out. I think this would be helpful for us to make sure that we fill this in. Which of the following terms can be used in the PNS, CNS, both? All right. So using what we got here on our page, and hopefully using the clues that you see up here on the screen. That's not cheating. It's called learning for now anyway. What would you say ascending? The word ascending. Is the word ascending kind of a CNS only word, PNS only word, or could it be used in both? Yeah, CNS only. Ascending, CNS only. It's my blue track that I got over here on my awesome ice cream cone. Okay. Next, motor. Motor. Can you use motor in CNS? Both. Yes, both. So in the CNS, the motor are called descending. In the PNS, the motor are called, motor. Motor or efferent. How about nerve? The word nerve. CNS, PNS, or both? PNS only. Peripheral only. Peripheral only. How about efferent? I just kind of gave the game away. That's the other word for motor in the peripheral. Okay? All right, again, so if you're following along in that left column, ascending, central, motor, both, nerve, peripheral, efferent, peripheral. How about tract? Tract, central only, central nervous system only. Tracts are the names for bundles of axons in the central nervous system. tracts are analogous to nerves in the peripheral nervous system. All right? Descending. The word descending. Central only, central nervous system only. And sensory. 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 Sensory is a both word. Sensory is a both word. And again, if the little exercise was a little confusing, go back up to those little lines right at the very top of page 132, and you can see before you filled in the blanks, it said, in the PNS, there are sensory and motor neurons. In the CNS, there are sensory and motor neurons. But in parentheses, they go by different names. Excuse me, different alternative names, I should say. Right? So neurons can do sensory, they can do motor, they can do integrative. I talked about that already. Nerves and tracts, talked about that already. C and PNS are both. I talked about that. We just did that. Now, okay, histology of nervous tissue there at the bottom, page 132. Sometimes I get ahead of myself and I forget that I have slides, for these things that we've already covered. Under the microscope, nervous tissue consists of two groups of cell types. And each of these has multiple different subtypes. But what is the general function of each one? What would you say, and some of you have looked at the video for this and already started to fill this in, but would you say is the general function of neurons? Conduct a nerve impulse. Perfect. I like it. Yeah. To create an impulse, but also not just create the impulse. The word conduct means to send it, to transmit that impulse, to send it away. Yeah. And use those impulses to communicate. The impulses are our body's major method of cellular communication. Yeah. To carry those impulses and transmit information from body tissue to body tissue and thereby control body function. Sure. But if we drill down very specifically to the function of neurons specifically, I would say conduct an impulse. Is probably about the simplest way we can start out. Now it's going to get more detailed, because we got to talk about what is that even? What even is that? An impulse. And we got to talk about what it means to conduct it, which means to carry it away, to carry that impulse to a distant location. Okay? Before we talk about that, we're going to talk about these support cells called neuroglia. Neuroglia. What is the general function of neuroglia? What's their general function? I kind of said it already, but I think you could put it in one word. Overall, what do the yeah. So I like support and maintenance. I like that. Support and maintenance. Okay, support and maintenance. Yeah, I like the way that's phrased. So we mentioned these back when we were talking about the histology of all tissues. I know you remember studying these. And one of the things that we talked about was of the different types of cells in nervous tissue, only one of the two types divides into tumors, and it's the neuroglia. Why is it that nervous system tumors come from neuroglia? What can they do that neurons cannot do? You guys remember that? Yeah, mitosis. So neuroglia are support cells. They also have the ability to do mitosis. I want to show you this on this slide. See if I can kind of show it in a way that stands out. Right in the middle of this photo, that's the nucleus, and there's the nucleolus. of this cell. And these branching things going out in all directions are dendrites. They're part of a neuron. This thing here is a neuron. That's a neuron cell. Okay? And that's kind of how they look. We'll look at the structure of these in just a minute. We've even got a nice little spot over there on page 133. We're going to draw one, whether you like it or not. Whether you like it or not, you're going to get to watch me draw one. Here's the thing I want to point out. Look at that. Look at this. Look at that right there. Look at these right here. Here. And all of a sudden you look around and they're all over the place. It's kind of creepy. It's like they suddenly start coming out from all over. These are neuroglia. These are neuroglia. These thin bands that you see right here, these are axons and dendrites from other neurons. But all of these cells in between, those are neuroglia. Those are support cells. It's estimated that the neuroglia actually outnumber the neurons by as many as 10 to 1. As much as 90% of the math of your central nervous system isn't neurons that we're going to spend so much time talking about, but it's the supportive neuroglia cells. Now, they come in a lot of different types, but if you'll take a look at the bottom of 132 and the top of 133, we've got 4 listed there that are part of the central nervous system, and 2 that are part of the peripheral nervous system. And if you look at them, which one of all 6 of those, starting with number 1 at the bottom of 132, going all the way through 2, 3, and 4, and then 1 and 2 at the top of page 133, of all of those, which one of those do we have the most to say about? It's the astrocytes. The astrocytes, a ton of research has been put into the astrocytes in the past couple of decades. For sure since 2000, starting in the 1990s. And it seems like at least every year, sometimes more than once a year, some research comes out of the neurology science literature that says something new about that we didn't know about astrocytes. They're multifunctional cells. They're all over the central nervous system. They do all kinds of things. They form what's known as the blood-brain barrier. There's a nice question there at the bottom of 132. What does that mean? What does that mean? Any ideas? What does it mean to say something like the word blood-brain barrier? If these cells form the blood-brain barrier, what does that mean they have to do with? What do they do? Yeah, the word barrier does make you think of separation. Yeah. Mm-hmm. It's right. You can sort of see it a bit right down here in the bottom right. Inside right here is a capillary, a little blood vessel. Yeah, that's a better way of thinking about it, Wanisha. It controls is probably a more accurate way of thinking about this kind of barrier. Selectively permeable, good way of thinking about it. Filtering mechanism, but it's a really active filter. It's more controlling. And it really controls what gets out of the blood and into the brain. There we go, Dong still got it. It really controls what gets out of the blood and into the brain. All of this space right here, if you guys can see where my little pointer is, all of this space here, this is brain tissue. And blood vessels have to come through because they bring oxygen and nutrients. But they potentially could also bring bacteria and toxic chemicals as well. So the blood-brain barrier, look what we actually have here. It's not so much a barrier as it is astrocytes completely wrapping themselves, covering the capillary. Like, except for where we got it cut right here, you can barely see the capillary. You can't see the capillary at all. It's totally coated in these little feet that stick out from the astrocytes. So they form the blood-brain barrier. Yep. They also, on the other side, they can attach to and support axons of neurons. They regulate the chemical balance around the cells, particularly potassium. Excuse me. One of the things that I want you to remember that we've talked about many times in the past is that all of your cells have what are called potassium pumps. Okay? And they pump sodium out and potassium in. What that basically means is the environment, because so much sodium is pumped into cells, the environment outside of the cells needs to be pretty low in potassium. That's going to be really critical when we start studying how these neurons actually function, what they do. And what they do when they function has everything to do with sodium and potassium. And to be able to work properly, you have to have a lot of sodium outside of these cells and very little potassium outside of the cells. So these astrocytes regulate that potassium of the environment around the neurons so that neurons function properly. That's a big flipping deal. That's a big deal. All right? They help to remove chemical neurotransmitters like the one that we talked about in the previous chapter, acetylcholine. They help to remove them. That doesn't sound like a good thing, but it is, because you don't want a signal from a neuron to go on constantly. What these actually do is help the signal go away when it's time for the signal to go away, actually. They help repair damaged cells. During fetal development, while your brain was growing and developing as a fetus, they even help to guide neurons and form connections in your little fetus brain. So they do a lot. Some other cells called microglia. You don't know what they look like in any of these pictures, but I like the visual diagram. Over here, kind of in the middle, are these tiny little cells called microglia. And microglia are actually a form of a macrophage. Is that why choline is important? Yeah, choline is used for a lot of things. It's not just part of acetylcholine, but yes, yeah, very, very important. Microglia are a type of immune cell, but they're an immune cell that's highly specific to the central nervous system. So you could almost think of them, I imagine them being kind of like a Secret Service that work inside the White House or work inside the Capitol or something like that. It's like they're not just, police officers. They're like highly trained at a very specific job and for a very specific location. So that will make a little better sense in second semester when you learn that most other places, macrophages kind of wander in and out of tissues and organs. They come and go, but these do not. These microglia are they come here to do this job in the brain or spinal cord, and they stay in the brain or spinal cord their entire lives. Up here in the upper right-hand corner of our diagram are these rows and rows of little cube-shaped cells called ependymal cells. Ependymal cells. The ependymal cells, it's going to be kind of hard to see this. I see now that they're really tiny on my screen. But right at their little tips, they've got cilia. Maybe if I let me draw on it and see if I can emphasize them a little bit, make them stand out. Right on their little tips, they all got little cilia. Oh, that didn't help much at all, did it? Lots of little cilia. And those cilia beat in a kind of a rhythmical pattern. Sort of like I'm doing with my fingers. They kind of beat in this rhythmic pattern. And they create a current that causes the fluid in the canal here to flow. They can kind of create this flowing current. And they also secrete the fluid that's in that space, in those spaces. And that fluid is known as cerebrospinal fluid. We obviously abbreviate that one. That's a big word that we're going to use a lot, cerebrospinal fluid, or CSF. So these ependymal cells make it and circulate it. I mean, it's not just increasing surface area. They actually move. So these are not like microvilli on some cells that just increase the surface area. They actually move and circulate the fluid through the space. Yeah, they're motile. They're movable. The cells create the CSF. The cells create the CSF, and the cilia circulate it. Yeah. It's easy to do. The last cells here are called oligodendrocytes, and the oligodendrocytes, there's one pictured here, there's another one pictured here. They stretch out and they wrap around the axons, and they form this layer of material around the axons that's referred to as myelin. So they actually form what's called the myelin sheath around the axons of the neurons. Now, those are the four that are found in the central nervous system. Out in the peripheral nervous system, we also encounter a couple of support cells. One type are referred to as Schwann cells. These are the ones that form the myelin sheath around the axons, but only in the peripheral nervous system, the PNS, the peripheral nervous system. So the Schwann cells of the PNS are kind of like the oligodendrocytes of the CNS. They kind of make the same thing. They do it in a little bit different way, and we'll talk about some of the differences between the myelin sheath of Schwann cells versus the myelin sheath of the oligodendrocytes in a little bit. But there's also these supporting cells called satellite cells, not pictured here. Let me sketch a couple of them. They usually are found near the cell bodies of the neurons. They usually kind of pack in between these dendrites. So they're found pretty close. They might be found kind of on the surface, on the outside, clustering around the cell bodies of the neurons, especially those that are outside the central nervous system. We're not honestly 100% sure what these cells do. They look like packing material. They look like packing peanuts, in something that just keep. But they probably have more of a supportive function that might be similar to. Well, I'm not going to speculate. You guys don't need to know. You don't need to know. They're supporting cells, and they are going to be analogous to astrocytes. The question in the middle of page 133. So Schwann cells are analogous to the oligodendrocytes. The satellite cells are analogous to astrocytes, the astrocytes of the central nervous system. They're most like the supportive astrocytes. They probably play a role in regulating potassium in the chemical environment around the neurons too. So let's take a look at the overall neuron structure. All right, now let's get to the neurons themselves. I had one pictured right here. It's this blue thing on that page. Well, let's draw one. Let's do it. I'm ready. I hope you're ready. You may have one already drawn. I draw one like this. I kind of make a U shape. We are in the middle of page 133, and there's not much space. You don't have a whole lot of space there. And I draw another little thing like that, and I draw another little thing like that, and I keep just doing that. At a certain point, I'm going to draw them together, kind of pull them together. I'm going to make something more like a tube, like this. And then This is going to branch out like this at the very end. It's going to have these little round buds that look like the end of a drumstick or something like that. Axon terminals. Just draw a few of those things like that. Creepy looking. Okay. I'm going to draw something like this. Put some chromatin in my nucleus there. Let's see what else I need. Oh, I'm going to need some of these. Doesn't have to be fancy. I'm not going to take too much time to do it. I'm going to need some of these. Kind of coming down right in here. I'm going to draw just maybe one of these inside this tube. These are little neurotubules. Now I'm going to switch up my colors here a little. And I'm going to draw one of these. I'm just going to draw something that's kind of like a rectangle with rounded edges. I'm going to draw another one not directly attached to the next one. Let's see how many. It looks like I'm going to be able to get 4 of them. So I feel like I'm Bob Ross. It's like talking while I paint. So I'm going to just going to paint a happy little tree right here, guys. Just a happy little tree right there. Look at that. Yeah, happy neuron. All right, so we got some happy little Schwann cells. All right, so I've got a happy neuron with a happy little Schwann cells. Let me label some stuff. Okay. Big things. This whole area here where the nucleus is the cell body, also called the soma. Perikaryon is the other term for it. Cell body or soma, either one of those words is okay. I just have gotten used to using the word soma, and it's just one word, 4 letters. But you can call it cell body, that's fine too. These shorter extensions coming out of the cell body are called dendrites. These little shorter extensions are called dendrites. And you can tell they're dendrites because they're kind of triangular, maybe they're kind of cone-shaped, and they get narrower as they get further from the cell body. This thing over here, this big long thing that I put over here, it's the axon. And it starts right about here, right about there, and it goes really all the way over to right about here where it starts to split up. And it doesn't taper off. It doesn't get thinner. It's more of a cylindrical shape. It's microscopic. It's tiny, but it's more like a tube. So it's more tube-like. The dendrites get narrower, skinnier, the further away they get from the soma. The axon, you can tell an axon because it gets a little skinnier right down here, but this area right here is the beginning of the axon. This region right in here, I'm going to kind of do a little dotted line to indicate it right there. That's called the axon hillock. Okay, it's sort of like it's kind of like the trunk of a tree, where the base of a tree kind of narrows, but then the tree itself is, the big trunk of the tree is like the axon. So the axon hillock is that. Way over here at the far end, these things are called axon terminals. Each one of these is an axon terminal. And at the very end, synaptic end bulbs. Synaptic end bulbs. The little rounded at the end. We looked at those in the last chapter when we looked at that thing on the muscle that looked like this, and it had the vesicles inside, and one of them was erupting like that. It was releasing acetylcholine. back on page 112. Back on page 112. What we were looking at there on page 112 was a synaptic end bulb. Synaptic end bulb. And those bulbs are the ones that have the vesicles with a neurotransmitter in the end of them. This thing that it was coming off of was an axon terminal. Right? But we didn't really see very much. We didn't see where it was coming from at that time. We just wanted to see what it was doing to the muscle that we were studying. Now we're looking at where that whole thing was coming from. All right? Now, I'll get to it. Okay. So, let's see. We got a couple of other things to identify here. These little things in here, in the cytoplasm of the soma, those little things are called Nissl bodies. Nissl bodies. Nissl bodies. Franz Nissl, the German researcher that kind of described those, and so they got named after him. I only mention that because it's a good idea to make sure that you capitalize that word, because it is the guy's last name. They're named after him, so it's his last name, Franz Nissl. Speaking of Germans, this is not necessarily on your list, but each one of these is a Schwann cell, and that's not pronounced correctly in German. It's Schwann. The W has a kind of V sound, Schwann. I think Theodor Schwann was the guy's first name. He lived at the same time as Franz Nissl. There was a lot of words in the 1800s that come from German that are named after people because they were the ones that had the cool microscopes and developed a lot of cellular structures. So these are Schwann cells. And collectively, all together, they make up what's called the myelin sheath. Okay, the myelin sheath is really all the Schwann cells together. And the little spaces in between them, are referred to as the nodes of Ranvier. Nodes of Ranvier. Louis-Antoine Ranvier. Not German. If I say it correctly and say Louis-Antoine Ranvier, where do you think he was from? If I say Ranvier correctly. Ha ha ha.